Welcome everyone, and to those joining us live, thank you for joining us today, wherever you are in the world. And for those of you who will be joining us afterwards, I'd also like to warmly welcome you and our guest to today's FIP event. My name is Chris John. I am the FIP lead for the COVID-19 digital event program in 2022. FIP's vision is a world where everyone benefits from access to safe, effective, and quality and affordable medicines and health technologies. Our mission is to support global health by enabling the advancement of pharmaceutical practice, sciences, and education. We're pleased to be delivering this event today as part of a 7-event program on COVID-19 this year. This is the 6th of 7 events and very much focused from the pharmaceutical science perspective. The program of events is called How Has COVID-19 Changed Us? Pharmacy and Pharmaceutical Science Perspectives. Pharmacists and pharmaceutical scientists still have an important role to play in the continuing response to COVID-19, as well as preparing for future pandemics. And as I said, the series of events has focused on therapeutic advances and change developments across science today, practice, workforce, education, and policy. So today's event is called Pharmaceutical Science, Reflections on Innovations and Collaborations During the COVID-19 Pandemic. We're very pleased to have our very esteemed and expert speakers today. And you'll see on the program here, we've got Dr. Juridal joining us from the University of Copenhagen, Professor Yvonne Perry from the University of Strathclyde, and we'll have a Q&A session towards the end of the event. So do please keep your Q&As coming in. We're hoping Dr. Adupa will be joining us, but we've been having some technical difficulties from his end, so we'll keep you posted about that. This is today's learning objectives. The aim is to reflect on innovations and collaborations in pharmaceutical science that have had an impact on COVID-19 and to consider how to apply this learning in response to future pandemics. And you'll see the learning objectives will show hopefully how pharmaceutical sciences have innovated and collaborated in the fight against COVID-19, challenges and opportunities in the development of medical products, approaches for fast-tracking diagnostics and treatments and vaccinations, and perhaps to reflect on what we have learned. Some brief announcements. This webinar is being recorded and live streamed via YouTube. The recording will be available on our website afterwards at www.events.fip.org. Please do ask questions using the question box provided, and we will hopefully be able to deal with those towards the end of the event. And you're welcome to provide feedback on this or any other event you attend to webinars@fip.org. Please do become a member of FIP if you aren't already, and the address for joining as a member is shown on the screen there. So it's my very great pleasure to introduce our first speaker, Dr. Louise Juridal, who is a researcher at the University of Copenhagen. I know Dr. Juridal will have a little bit of introduction about her work as in her presentation. She's also a regulatory scientist, and her research interests include challenges related to regulations of medicines and medical devices and regulating emerging technologies. So a warm welcome, and the floor is yours, Dr. Juridal.
Thank you so much, Chris. Thank you for the kind introduction and for the invite to speak. And hello, ladies and gentlemen. Been looking a lot forward to today and to take part in this FIP event. So I'm happy to present to you on some of my work that is with regards to collaboration in times of crisis. the COVID-19 vaccination research and development partnerships that I have been conducting in collaboration with Professor Timo Minsen here from University of Copenhagen and Professor Nicholson Price from University of Michigan. So very briefly about myself, I'm also a pharmacist, and then I did my PhD in regulatory science. Since then, I have taken a more journey towards regulation and law, and I am now based as a postdoc at the Faculty of Law at the University of Copenhagen, where I have been working with biologics and manufacturing of biologics. And at the moment, I am on a project about, the classical project on AI-assisted surgery and the regulatory legal aspects of that. But for now, I will like to tell you more about what it is that we should talk about, or I want to talk with you about today, and hopefully we can discuss as well later on. So the agenda for my talk is that I would like to introduce you to some types of innovation ecosystems, more concretely about this study that I have been conducted together with Timo and Nicholson, and also what possible challenges or learnings there are on the type of partnerships that we have seen during the COVID-19 pandemic. So we just look briefly overall on the types of innovation systems. We can overall talk about, at least according to these authors, about 3 types of innovations or ecosystems. So the first one is about business as a business ecosystem, which focuses on direct business benefits and to create customer value. There's typically the economic outcomes and the business relationships between actors that are highlighted. Then there's a talk about a knowledge ecosystem, which aims to generate new knowledge and technologies. And that is also with regards to an interest and outcome in creating new knowledge through joint research work, collaboration, and development of knowledge base. And then the third type is innovation ecosystem, which integrates exploration of knowledge and with exploitation of this knowledge in a business context. And this also includes mechanisms and policies that foster creation of, for example, innovative startups. And for in this case, we will be focusing more on the knowledge type of ecosystem. So the study that I have made is on the collaborations that were to develop COVID-19 vaccines. For this study, we used the WHO list of COVID-19 vaccines under development. However, we did last extract the data in May 2021. From this, we saw that there were actually, which I was quite surprised about, but there were actually 283 vaccines either in clinical or preclinical development. We then found that, so looking at these, there were 99 either in clinical and 184 in preclinical stage at the time. And then we analyzed which of these, like how these vaccines were, like who were the sponsors of these vaccines, and we analyzed whether these were entities, or so it was singular entities, or it was a partnership of 2 or more entities together. And from these, we found that by large, it was mainly single entities developing COVID-19 vaccines, because there were 190 of them, and 93 of them had 2 or more entities. However, so when we after this, we characterized the partnerships. We looked into what type of partner took part in the partnership. So it was an academic partner, a company partner, governmental partner, and etc. I will show you more on the next slide. And we also analyzed which type of generation vaccine technology that was used for the given vaccine. And subsequently we tried to, we found out what type of partnership that was. We'll introduce you a bit more in the next as well. However, 12 of the vaccines were excluded. So if we look a bit more to the next, then we look at the figure starting on the left side, then we can see the 190 of them. On the yellow part was the singular entities, and then in the bottom we can see the 81s that were conducted by 2 or more developers. So if we then look and see how many of the 81 that were conducted by 2 or more entities, that will result in the figure then on the right side. We can see that actually of the 81, then 71 of them had a company partner involved, which is quite a large percentage. And if we look at all of the ones that were conducted by 2 or more, we could see that actually 56.8% had so-called materials transfer partnerships, which are, so it's transfer of materials, it's technical infrastructure or IP rights transfer. Typically, that's a type of collaboration that follows a model of serial innovation steps. So one partner would pursue early innovative steps and then transfer the resulting materials, technical infrastructure, and intellectual property to another partner to pursue further steps. However, there were also 43.2% that were involved in so-called knowledge sharing partnerships. And those we characterized as partnerships that were involved with sharing of knowledge and technological expertise, such as if the 2 partners have complementary expertise and information that is collaboration with regards to information in an ongoing process to develop technology and crucially to increase future knowledge and capabilities of both partners. And then if we look a bit more closely looking into, so what type of, I don't know if you can see my mouse actually here, but so if we look then a bit closer into what type of partnerships is it then that are undertaking these 2 different types of partnerships. We look on the table 1, so on the left, and we can see that actually, so there were 35 of partnerships that were involved with sharing of knowledge and 46 that were involved in transfer of materials. So we can actually see that there were more collaborations with focus on transfer materials rather than exchanging and increasing knowledge of both partners. If we look, then it was by far mostly either company-only partners, but or if there was an academic partner, and to some extent also governmental partner that were the most frequent partnerships. Only to a limited degree were there partnerships that did not have a company partner, and also to a limited degree partnerships that involved either a hospital partner, military partner, or non-for-profit but private organization. It is interesting to look to see that there is quite a large percentage of the partnerships that involved an academic partner. If we look a little closer to that, actually, we see the figure on the right. So for this analysis, we used Real et al.'s definition of either classic vaccine technology, which include whole inactivated virus, live attenuated virus, protein subunit, or virus-like particle. So either a classic or alternatively a next generation vaccine technology, which included viral vector, DNA, RNA, antigen presenting cells. And if we use these 2 definitions together with a subset of the data, which were of 2, if it was a partnership with at least 2 developers, whereof one of them must be from academia, we could see that actually there was quite a number more where academics were, or an academic partner were involved, that where there was knowledge sharing that was also related to a higher use of next generation vaccine technology, comparing to if it's a partnership with an academic partner that is undergoing material transfer type of partnership, then was more frequently with a classical vaccine technology. So we then focus a little more into if it's since there was such a large percentage, it was 71 out of 81 partnerships where there was a company partner, we can try to zoom out a little and see what are the common challenges to public-private partnerships between industry and academia. And there is a survey conducted by Gersthoff in a Nature review from 2019, saying that actually about 79% of the challenges that occur between industry and academia were due to either research constraints, which is type of challenge that involves either limited availability of human, monetary, or organizational resources, for example, tools or instruments for the project. The other type is coordination challenges, which relate to difficulties in teamwork, frequency and quality of communication. It could also be the coordination of tasks on exchange of goods and knowledge. And the third types of challenge would be legal administrative process, or so-called more like paperwork challenge, which could, for example, arise to internal approval processes, ethical reviews, contract negotiations, take time and or can limit a partner's access to relevant knowledge. And the fourth type of challenge is related to scientific challenge, which could be more related to, for example, negative results. It could be issues with scientific methodology or difficulties in interpreting data. And if we try to, for a moment, then to think about, okay, so these are the partnerships that we have seen during the pandemic period for developing of COVID-19 vaccines. We've seen that there were a lot of them were like a lot of innovation were spurred from this. But there were also clear incentives for knowledge sharing, for collaborations during the period. And one of the things that I find very interesting is to look into, okay, so now we're, even though COVID-19 and Corona is not over at the moment, if we then look a bit beyond this situation and considering a new non-pandemic situation, then what is actually realistic in such non-pandemic situation? What can we learn from this, and what incentives do we have now? Because I think we had a very rapid development of vaccines that were, I think, prior to the pandemic, not something that anyone would have believed possible. And it's interesting to see how we can actually take some learnings from that and see what could be used in also more everyday not vaccine, but medicine development. And also a part of this to say, well, how do intellectual property and knowledge sharing actually play a role for generation of new knowledge and technologies and collaborations? Those are my references. And then I like just to say, I hope you have some questions, and please use them in the Q&A tool. Thank you so much.
Thank you so much, Dr. Trudal. I was very interested by your figures there, and a large number of vaccines in development and quite a large percentage of partnerships involved. Let's hope it continues going forward. Thanks again. I'd now like to introduce our second speaker, actually, Professor Yvonne Perry, who's Professor of Drug Delivery at the University of Strathclyde in Scotland in the United Kingdom, and who undertakes research multidisciplinary and focuses on developing nanomedicines to facilitate the delivery of drugs and vaccines, providing practical solutions for current healthcare problems. Professor Perry is a member of the Academy of Pharmaceutical Sciences and has extensive experience across academia and industry. So I will hand the floor over to Professor Perry, and I will just stop sharing because I think Professor Perry is going to share her slide.
Yeah, lovely. Thanks for that, Chris. Let me just get everything set up. Okay. Presentation. There we go. Chris, could you just confirm that's working okay?
I can't see the slides at this moment.
Yeah, it's been stopped due to external display has been disconnected. Oh, can you see now?
Try sharing again.
Okay, yeah.
Go through the screen share again.
Yeah, I'm going to do that again. Okay.
That's all great.
But am I still on present? Can you see my notes or the full screen?
We can see the full screen. That's perfect.
Okay, perfect. Okay, we're good. Excellent. Okay, thanks for that, Chris. So yeah, following on from what we've just heard, so I'll be talking a bit more about the challenges and opportunities that we've actually seen. So we have seen that therapeutics and vaccines were developed in response to the pandemic. And we had a range of vaccines. So we had our viral vectors, so our Oxford AZ and our Johnson & Johnson came through. In terms of the RNA, which are probably the most widely known now, we have the Pfizer-BioNTech and the Moderna. But we also have whole virus vaccines, so Sinovac, and there's a subunit, Novavax. In terms of antibodies and antivirals, so for the therapies, we had our neutralizing monoclonal antibodies developed, and we've had antivirals. So we've had a range of those. Paxlovid is the most probably common people know now, but you can see both angles were developed during the pandemic. But I'm going to focus mainly on the vaccines. So in terms of the mRNA vaccines, we had them first approved in December, and then they have gone through an evolution. So the Pfizer-BioNTech was the first to be approved. We had the very first formulation that had to be diluted, and at the time the storage requirements were quite strict. But as we developed more information and generated more stability data, we were able to extend that shelf life. And if you look across the screen, basically you can see that basically we've had a change of various types of vaccines. So we've added different age groups, we've added the ability to have them ready diluted, so there was no dilution, and that's applied to both Pfizer and Moderna. And basically the key thing for a pharmacist or a vaccinator was this cap colour. So at the moment we're working with the bivalent, so both of them have some elements of grey, so the Pfizer is a grey cap, Moderna has a grey outline to it, but basically they have both the original Omicron, and now encodes for the Omicron, the new variant. So, and they've split the mRNA content, so it's 50% of each. So we can see the evolution of these has been going on even since their development originally. So the question is, how did we manage to achieve in a year what used to take us 20 years? So it was very well accepted that a vaccine would take about 20 years for us to develop. And that's because there was various stages. So as shown here, and this is actually from the EMA, they have a very nice website on this, but we start with small scale studies, we progress to non-clinical with our in vitro and our in vivo, we then move to our clinical trials, which we expand, we then have evaluation decision by whatever regulatory body covers your country, and then we have, once you've had it approved, you scale up production, commercial managing, and then we have our pharmacovigilance or our safety monitoring that goes on after the launch. So you can see why that would take so long. So how did we achieve this in a year? And that was one of the key questions. People were concerned about how it was done so quickly. So you can see the timelines here. So the blue is what would usually be done, and then the red is what we did during COVID. So the first thing to see is in the standard process, one phase follows the next. So there's very little overlap until you get down to the production. But if you look at the COVID vaccine response, what the regulators and the manufacturers did was to overlap each of those areas. So a lot of things were done overlapping, hoping that things would work, but if not, they were running maybe 2 or 3 streams at once. So it was achieved in 12 months, but also it maintained the safety, quality, and efficacy. So you can see how overlapping these streams now can make it quicker, but how is that feasible? So if you look at what we had, if you remember, it's lockdown, it's pandemic, people really just focused on this. So normally you would have your developer, so the company working with your regulator and scientific experts, working together. Once it's approved, you would go into production. But if what happened in the pandemic was basically there was 4 things. So in terms of development, they applied extensive previous knowledge. So they brought in lots of people, both in the developer side and the regulator side. So they put a lot more resource in. And the second point, resource was mobilised to look at this simultaneously. So they were looking at various parts simultaneously. And they had this constant dialogue. So the regulators, wherever we were, and the developers were working hand in hand throughout. And they had a much bigger team than you would expect. And then the fourth element of this was the manufacturing. Manufacturing did start earlier on the assumption it would be approved. So one would say this would be a financial risk, but as long as everything checked out, then we had production. And that was able to do this because we had to do it during a pandemic. People were willing to work. I mean, there was 3 rotations in many of the companies working on shifts. I know the regulators across all the areas were working extremely long hours to make this happen. So we now have this assumption that vaccines can be approved in a year. And that actually presents as a challenge because you have to manage these expectations and we have to manage resource because we won't be able to continue to put that amount of effort into something and compress all that work into a year. So it is about managing the resource, managing expectations, but also taking what we've learned from this pandemic and what we've learned from the vaccines. And that switches into an opportunity. So if you look at our mRNA vaccines, they really could be considered as a platform technology. You have a lipid nanoparticle encoding mRNA, and the idea is you can swap in and out. So is this an opportunity for us to scale these up and produce them and approve them quicker? So that's the things to look at with these mRNA. So what is next for us and where are our opportunities? So in terms of manufacture, we do want to look at scale independent. At the moment, there was a lot of work done. We scaled out, the companies scaled out rather than scaled up, and other people would then scale up. So the idea is, can you produce 1,000 batches, 1,000,000 batches, all in the same platform without changing your process? Can we add an inline process assessment so that we're not doing batch release? That would speed up things. And finally, global accessibility. If we can manufacture in various sites rather than manufacture in one site, if we can distribute manufacturing, access will improve. The next part is we can look at our payload. So we know they work for mRNA, but what about the smaller RNA? There is one product approved. We can exploit this much more. And also self-amplifying RNA. And I'll speak a little bit more about that opportunities. And then in terms of what we could look at to improve efficacy, so we can look at different ways to deliver it. We can look at alternative to lipid nanoparticles, could be others. And could we look at targeting to improve potency? And then finally, administration. Can we look at a needle-free route so that it could be potentially deployed to our households in future so we can self-administer these vaccines? So these are all potentially what would make the next steps. So if we look at what is a typical lipid nanoparticle, so basically it's a combination of 4 lipids. So we have 2 stabilizing lipids, distereophosphatidylcholine shown in purple, and cholesterol in the yellow. That makes up about 50%. We then have a ionizable lipid that has a positive charge when it's manufactured that binds to the mRNA. And then we have a small amount of pegylated lipid that stabilizes these nanoparticles. And in terms of manufacture, most of the time it's based on basically a dilution process. So you have your mRNA in an aqueous phase, you have your lipid in the solvent, and as you dilute down, the lipid forms into a nanoparticle. And we can use 2 different types of microfluidic mixers, and we do this generally in our lab. So we tend to use a staggered herringbone mixer or a toroidal mixer as shown. And this ability to microfluidic manufacture gives us scale-independent production from bench right up to GMP. So what you can see here is our production in our labs. So we have the small staggered herringbone mixer that runs at 12 mils per minute through to a GMP machine that runs at 200 mils per minute. Now the green bars are the particle size and the white dots are the polydispersity. And what you're really wanting to see is everything matching so we can jump to bigger and bigger sizes, bigger and bigger production without impacting on our particle size. And equally, we want to have the same amount of mRNA entrapped. So we've sped up production from 12 ml a minute up to GMP, 200 ml per minute, and we're seeing the same product. So we're able to move between these productions. So we can have scale independent manufacturing using microfluidic technology. But what about payloads? So you get different types of payload. So a lot of studies now we're using poly-A, which is the short tail. It's very small, but it's very cheap, and it allows us to do a lot of investigation at a low cost. But then we can have mRNA and we can have self-amplifying RNA. So self-amplifying RNA basically contains genes of the alpha virus, and it allows us to encode for non-structural proteins that improve replication. So basically, it self-amplifies the RNA, and you can dose down. So you can use much less dose, so it reduces cost and hopefully improves our access to these products. So we've been doing some work in our labs, and we work a lot with mRNA and self-amplifying RNA. And basically what you can see here is we have made a range of mRNAs, and we've got a range of self-amplifying RNA. And again, the bars are the particle size and the dots are the dispersity. So you want the same size and low PDI of dispersity. But what you can see is we can load mRNA and self-amplifying RNA that encodes for different proteins. It always gives us the same loading and the same encapsulation. So the point of this is we're showing that this is a platform technology. You can use these LNPs, you can load in any RNA or self-amplifying RNA, and you still have the same package. So then we looked at comparing mRNA and self-amplifying RNA, and we put them in two different types of LNPs, and basically we dosed down. So with self-amplifying RNA, we'd use 1/5 of the dose. And what you can see here is we have 5 mice immunized with mRNA and 5 mice immunized with self-amplifying RNA, and the fluorescence is the protein expression. So with the messenger RNA, you get very early expression. It comes up and goes down, whereas the self-amplifying RNA comes much slower. And if you plot that in the graph, this is what you see here. So the mRNA is high and then decreases, self-amplifying RNA takes a while and then comes up. And that's made with 2 different LNP formulations, and it's a similar process. And what it's not related to is clearance of the LNPs. So the pharmacokinetics of these particles are the same, irrespective of what mRNA or self-amplifying RNA we have in it. So again, this is us showing that we have a platform technology with these LNPs. They can deliver different types of RNA. You can control the pharmacokinetics of expression depending on the type of mRNA, but it means that you can use this again as a platform technology. And this is us showing us that we made these formulations encoding for COVID. So these are actually COVID vaccines that we tested in mice. The mice were injected at 0 and 28. 2 different LNPs formulations, both working very well. And this is at a 5 times lower response than we would normally use for the mRNA. So we've dosed down and we've used different platforms. So again, self-amplifying RNA lets us use less mRNA. And we've got types of LNPs, so you've got different platform technology. We can also use a range of nanoparticles, so not just LNPs. So again, this manufacturing process may offer new opportunities for different types of LNPs. So these are process parameters. You can see it's very similar. And we're always getting particles below 100 nanometers with very high loading of our RNA. So that's great. So we've got a manufacturing process that can be scale independent. It's deployable. It's suitable for a range of nanoparticles. And finally, that last idea of can we get rid of needles? Less about needle phobia, it's more about the clinical impact of all those needles and also the deployment. We saw it takes time to deploy a vaccine for a pandemic. We had to work through the age groups. Is there an easier way to do it? So we looked at testing various nanoparticles via different routes: intramuscular, intranasal, and intradermal. And here's our data. So this is actually for a rabies vaccine. So you're looking for immune responses. So intramuscular, we see good immune responses across the different nanoparticle platforms. And then the blue Rabipur is the commercial product. It's not an mRNA vaccine. It's a subunit, but basically you have to beat that, which they all do. So intramuscular works great. Intradermal, not much use for human really, unless you're looking at that, but again, working well. But the key we wanted to see was intranasal. So after 4 weeks, we'd administer these intranasally, very low response. If we go to 6 weeks, so this is 2 weeks after the second dose now, you have to check all the doses are tenfold higher now. So IM working great, intradermal working great, intranasal still very poor responses. So what this tells us is that this platform technology works intramuscular and intradermal, but we're going to have to reformulate it if we want it to work intranasal. So we can't just use the same vaccine format for a nasal route. We probably need it to be more mucoadhesive, residual time longer. So in conclusion, we do now have, thanks to the pandemic, a flexible platform technology for mRNA. We need to work more on our global accessibility. We need to bring down cost and have deployable manufacturing. And the third thing is we can now harness targeting technology because we have our base formulation. So let's look at the next generation of LNPs. And with that, I'll hand back.
Thanks so much, Professor Perry. That was an excellent presentation. Very exciting to hear about self-amplifying RNA, which is something I've learned today. And hopefully the other routes, such as intranasal, will take off a bit more. Although I used to be needle phobic, but I think I've overcome that now. But there is still a lot of people out there who are quite needle phobic, aren't there? And we need vaccine coverage to be as high as possible. So thanks so much. What I think we'll do now is just have a look at the Q&As we've been receiving, and if our panelists are able to answer, that would be fantastic. But just the first one we have is, are there vaccines that are being developed to combat newer strains of the COVID virus? Shall I go to you, Professor Perry, first?
Yeah, I can take that. Absolutely, yes. So we saw the bivalent that's now being used in many countries is actually the original Omicron and the new variant. And the idea is you basically snip out, well, that's what they did. They snip out the section of mRNA that they don't need anymore, and then they put the new one in. At the moment, though, this is not seen as platform technology, so it has to go through the approval process again. But if we can get more knowledge and more confidence with these things, then hopefully at some point it will be like the flu vaccine, which isn't reapproved every year. It rolls through. But yeah, definitely they are working on it. And I see Louise nodding her head. Louise, do you want to add something?
I'm just agreeing.
You've been monitoring the WHO. They have the full list, don't they, of everything that is being developed. So that's obviously a growing list. I think the New York Times as well does quite a good monitoring of all the vaccines in development as well. Although that's an unexpected publication to do it. It has emerged over the pandemic, certainly. Great, thanks. So just have a look at the other questions. One concern is, of course, how affordable would vaccines be for developing countries like the Philippines? Perhaps if I could go to you first, Dr. Duridal, what have you got a take on this?
I can give you my best, at least. I think it's a difficult question because I think so access and affordability has been a large part of the discussion during the pandemic, and unfortunately, I don't think that part is over yet. While I have some thoughts on access in general, I'm afraid I do not know the situation in the Philippines that well. But I think that there are companies also looking to deploy manufacturing more locally. And I think that hopefully will have an influence, but I'm not aware of any major new developments with regards that will make a dramatic change to the affordability we have at least seen so far for the already existing vaccines.
Thanks. And Professor Perry, do you have any thoughts on that one?
Yeah, definitely. I think it is something we do need to drive the cost down. So the Oxford AstraZeneca and the J&J one is much cheaper and has a better shelf life. I mean, we are now addressing that with mRNAs, but the price is still too high, and and that's the complexity of the manufacture. But hopefully, as we gain more experience, we'll drive that price down for sure.
Great, thank you so much. And we have a question on what are the future research opportunities based on, I think this is related to current outcomes related to COVID. That's quite a big question. Professor Perry, any thoughts?
Yeah, I think we've learned a lot, both on the therapeutics and the vaccines. Vaccines is my area, but if you look at what we've learned from the antivirals as well, and actually a therapy is almost better sometimes than a vaccine, because now if they've been vaccinated, you still can have symptoms. So the therapeutic treatment is vital. And I think we've learned a lot from what antivirals do work and what we can be crossed over and what hasn't. As I say, I can't speak in detail about that, but I think that's a really growing area, the antiviral, making sure we have those therapies available for those that need it.
Thank you. Yeah, we talked a bit about antiviral treatments in an earlier event, and it does feel like it should and can be a growing area, which to keep an eye on. Dr. Derrida, have you come across any research opportunities or developments from your research?
I think mainly there's a larger and larger focus on knowledge sharing and how that has played an incentive during the pandemic, and how we how that may not always have been the case otherwise. But if there are perhaps be a larger focus on policy intent, like initiatives, for example, also with regards to contracts that are or prices set, which could favor these things, at least some policy suggestions I have seen based later on the research.
Were there any trends you noticed in your research as far as product development was concerned?
So of course, the products that are mainly covered in media and so on were of course also in the data set. I think what we can see is that there was, of the 283 of them, it's fairly limited how many of them we have then seen as marketed products in the end. While of course there might be some of the ones that were on the GIA and WHO's list that are not being brought to attention outside of maybe the respective country where the development did occur. Yeah, I think that's in the publication. It's also you could see, of course, the pattern in it was, for example, the AstraZeneca was a very clear example of a materials transfer. It was clear that it was Oxford primarily who developed the vaccine, and then they were looking for a commercial partner to make it into a global product and to know about the regulatory procedures and market access and so on. Whereas we found that it was at least more sharing of knowledge that were undertaken in the BioNTech and Pfizer, even though it was, for example, it was more like unidirectional with regards to, for example, the funding that they received from the US government. However, the US government were not having any rights over the technology or knowledge from it. So there is still some details in that, of course.
There's been a lot of government funding across the world that has certainly helped from what I've seen. But, I think that may change going forward with the current economic situation, but who knows? This is potentially a good question for you, Professor Perry. What would be the main hurdles for intranasal formulations? And I think this is a question from Dennis Dubois. Is it adhesion to mucosa or MP stability in the special septic environment?
Yeah,
a very specific question, but it sounds a good one.
Yeah, but I think so far we haven't managed to have them reside in the nasal cavity long enough, I think. So for that study we did with the mice, we actually tracked the particles and it ended up the mice swallowed most of it. But mice are nasal breathers, and our formulation wasn't designed for intranasal, because the kind of research question we'd sent is, can you just take these vaccines and use them a different route? And the answer is no, you can't. But can they be reformulated to work and stay in the nose longer and give you an immune response? The answer to that would be probably yes. And that's, I think, the next step. And that would be an opportunity for people with more experience in the nasal route, I would say, definitely.
Great, thank you. Thanks for all the questions from our audience. I've just finished off with one from me. I think I was really struck, Yvonne, by your presentation about what an astonishing 12-month period it was that actually everyone with a Herculean effort got the vaccines developed and together. And I suppose my question is, learning for future pandemics or even future important developments, what is sustainable going forward as from a collaborative point of view? And what is the most important thing you think, and what is sustainable?
Mm-hmm. Yeah, I think that's a very good point, because we couldn't do this every time, because it wasn't, as you say, it was a Herculean effort. We all wanted to fix the problem, and there was an element and there was nothing else for us to do because we were locked up. But that's maybe a bit cynical. But yeah, I think there's a lot we can learn about, particularly the rolling review programs. So most of the regulators adopted rolling review. They would take data at different times and do that. So they were assessing it as it went, rather than wait until they got the full package. And I think with experience and confidence that we can do this can be implemented more. It does take a little bit more work because you are looking at different things at different times. So I think that, and also what we heard about Louise, more of the collaboration, and hopefully, and I saw Louise lovely summarised the hurdles, and one of them is, trying to get that contracts. I can't remember exactly what you had in your slide, Louise, but it was about the contracts and trying to get us cross-talking. And I know that can slow things down. I see that at my university, and I know other universities are the same. So I think that's something we could work on more and adopt a much more collaborative outlook, which we did for COVID, but I worry we'll regress back to where we were.
Yeah, that's a good point, and that is a worry. Dr. Drudal, do you have any perspectives from you on that one?
I think that definitely we need to I think a large part of it is, as I agree completely with Professor Perry, and I think a large part of going forward will be to try to lower the cost and look at what we can do to increase access going forward. Thank you so much.
We've been joined by our colleague, Dr. Adupa from India. And can I just test your speaker, Dr. Adupa? Can you say hello? Just see if we can hear you. We're not hearing you. That's a shame, because I was going to ask you for your perspective. Seems we're still having some technical difficulties there. So I'll have to perhaps move on from the question and answer. Thank you to our panelists for your expertise and dealing with all of those questions. It's really very useful to have your perspectives on this particular area. I'm just going to wrap up the event. It's been very useful to hear where we're at with science and what we've learned during this pandemic, how we've all worked together, and hopefully, fingers crossed, this will really give an impetus for future collaborations in any future pandemics, though I'm hoping we don't get too many too soon, but that we are prepared and we're ready. So this event has been recorded, and if you want to hear it in more detail, it's available at events.fip.org, as are all our other COVID-19 events and all of FIP's events. So it just leaves me to say thank you so much for attending. I hope you've enjoyed the event, and I wish you a good day.